from Malaysia. I'm sure some of us are at different time zones at the moment. Regardless of that, welcome to our fun international webinar series. The purpose of this series is to provide a medium for researchers and exchange views on any research topic. Before we get started, I would like to inform you that this session will be recorded. And if you refuse to be recorded, you can watch this session in our YouTube channel, that is F.U.N Lab. And the link will be shared in the chat box. So before we begin, here are a few housekeeping announcements. That is, please fill in the attendance form that will be provided by our admin for e-certificate. Please mute your mic while the presentation is ongoing. You may type in your questions in chat box, and I will only read them after the speaker has completed his presentation. So today we are grateful to host an exceptionally outstanding speaker, Dr. Won Young Lee, to share with us an interesting and, if I may add, quite a cute topic, which is penguins. The title of the talk is Behavioral Ecology of Chinstrap and Gentoo Penguins at Narebusky Point in King George Island, Antarctica. To begin, I would like to invite Noor Adilazani, our program coordinator for this current webinar, to introduce our speaker.
Okay, hi, good morning to all. Annyeonghaseyo, Dr. Lee, and welcome to our 8th webinar. So it's a great pleasure for me to introduce our speaker today. I get to know our speaker today in February 2020 after being introduced by Dr. Chin. He helps me in identifying the penguin species only by looking at the image of the bone of the penguin. How awesome, right? And how expert he is in that field. Today, he will talk to us about the penguin behavioral ecology in Antarctica. This is a subject we should all be deeply interested in because it's rarely been discussed publicly. Our speaker spent his career in the Division of Polar Life Sciences Korea Polar Research Institute in Incheon, Korea. He completed his PhD at Biological Sciences, Seoul National University in 2014. He also received a postdoc fellowship for future generation from Korean Research Foundation. His latest publication titled Interspecific and Intraspecific Competition of two sympatrically breeding seabirds, chinstrap and Gentoo penguins at two neighboring colonies, published in Animals on 11 February 2021. So everyone, please join me in welcoming Dr. Won Young Lee. Please, Doctor, the program is now yours.
Yeah, thank you, Adela. Good morning. Today I'm going to present my study about penguins. And my initial title was about Gentoo and chinstrap penguins, but while I was preparing my talk, I decided to enlarge my topic based on my recent publications. So maybe I can talk a little bit more about other animals, including like weather seals or other flying birds called skuas. So I hope you understand about my decision. Yeah, so let me start. I'd like to start in my background, my personal background. I was a little kid loving animals and flowers. Since I was young, I was very obsessed. In the zoo, I really liked watching animals, and also I really liked dragonflies. So I used to take my personal net everywhere, and I caught a lot of dragonflies. That means I killed a lot of insects. So I totally regret what I did at the time. So like many other kids, I was the kind of animal-loving guy. But since that time, I was not very changed. When I was a middle school and high school student, I still loved animals, and I still dreamed something about doing this in the future as a job. So in Korea, when you become a university student, in the first year, you usually go to Europe or America as a backpacker. So many of my friends went to France or England to the museum or other squares. But in my case, I was not interested in going to Europe. I was interested in going to Africa for safari. So my first foreign trip was the East Africa, from Kenya to Zimbabwe. So I really love to watch giraffe, wild beasts, and flamingos in the wild. So I knew that I really love watching animals, and at that time I decided to become a researcher, watching animals. I want to do this as a job. So now I became the one who is doing this as a job, and annually I go to Antarctica and also to the Arctic area. So especially in the summertime, I go to Norway or Greenland, and in the wintertime, I go to the opposite side, Antarctica. So since 2014, I've been there. I've been to Antarctica for 7 times, and probably this coming winter, I'm also supposed to be there, watching penguins and other seals. So when I say something about Antarctica, people used to say that, oh, that's cool and that's very fantastic. But to be honest, it's not very fancy because in the field, I am a really bad guy to the penguins, because I'm always taking a net, as I did when I was a kid, to follow dragonflies. So now I'm following the penguins, and I'm taking some blood samples, and I put some GPS devices, the trackers, to the penguins. So I'm pretty sure that penguins don't like me. but I'm really a penguin lover, but I understand that they don't like me because I am not feeding them, and I'm doing something really, I'm giving some stresses to them. So in the field, I'm always trying my best effort to reduce my job, and I'm always trying to find a way to reduce their stresses. So I believe that my research is not giving them too much stresses, better than the old ones. Old researchers just catch them, and sometimes they kill the animals to find something inside them. But I'm not doing that kind of very harmful ways. I am using some kind of devices on their feathers for a few days, and then I retrieve them very quickly. So I hope that you don't worry about that too much. So let me start what I did in the field as an observer. The first task was how to discriminate which one is females and which one is males. For humans, we are not able to distinguish them in the field. So, do you have an idea which one is a male and which one is a female? Yes, it's quite difficult. So because the phenotypes are very similar to each other, and their parental roles are also the same. So for penguin biologists, it was always a difficult task. But quite recently, of course, you may know that molecular sexing method. So if you use the specific area in in their sex chromosomes, then you can easily sex them. But in the Antarctic research station, it's quite difficult. So I made some very simple formula to distinguish them in the field based on their bill measurement. When you see them very, very carefully, then you can recognize that males have a bit longer and a bit broader bill length. So based on the bill length and bill depth, you can approximately distinguish them. It's about 90% of accuracy. So it's not bad, I think, to do this in the field. So I made the kind of formula, and based on my measurements, I easily sexed them. And when I was planning to deploy some GPS to males, then I can select them based on their bill measurement, and then I can select males. And when I want some females, then I can use the bill measurement for the females. So it's quite easy, but yeah, it's a basic method for the field biologists like me. And the second method is also very simple, just watching them annually, like seasonally. So on the left, you can see that weather seals are having pups. So you can see some blood. And recently, I became a father of 2 twins, and as I know, Dr. Chin also became a father. So I became very special when I see this kind of moment. But for Antarctic biologists, we still have not much knowledge about their breeding cycle, because Antarctica is quite a large area. So along the Antarctic continent, we need some accurate records about their breeding cycle, when they start breeding and how the breeding cycles are changing, probably due to the climate change and global warming. So in the Korean Antarctic Station, the overwintering members are recording their breeding cycles annually. So based on their breeding cycles, I reported that when they are having birth to the pups. And also I found a twin female, twin female. It's quite occasionally happening, but seals usually have only one pup. But very occasionally, sometimes they have 2 pups at the same time, as my wife did last time. So I felt that it's quite special, and so I recently reported this event, and I calculated that this twinning rate in the seals. So compared to the humans, seals are having very low chances of twinning. And also I reported that weather seals are hunting adult penguins sometimes. The weather seals are they are not very aggressive. And I think Dr. Chin observed weather seals in the Antarctica, but we did not care too much about weather seals because they are just sleeping on the snow, and sometimes they go to the sea and then they hunt fishes only. But sometimes we found that when they are really hungry and they can easily follow the penguins, then in that case, then weather seals are having penguins sometimes. So I think it's a quite recent changes. So because recently we are having very rapid changes in Antarctic ecosystems. So I think this kind of relationships are quite changing a lot. So I think that this is a quite rare case, but I think it can be a little bit more common in the future. Also, when I visit the Arctic area, I become a bird watcher. So I'm taking a binocular, and then I walk the ground all the time, from the morning to the night. So I photographed the birds species, because this area is the North Greenland, and the latitude was about 83 something. So it was quite close to the Arctic, the very high Arctic. So there have been almost no records about the animals. So I think I was the first one who camped there and made very detailed records about the breeding birds. So based on my records, I reported that there are many birds which was not expected to be there. Because previously, in this picture, you can see that Lapland bunting. You can see Lapland buntings and king eiders. But previously, people believed that they are in the Norway or Netherlands, in the West European countries, and so they are supposed to be a bit lower latitude. But quite recently, Arctic environments are also changing. So this kind of migrating birds are expanding to the north. So as a result, I think I could find some very new species in the high Arctic area. And it's a bit different topic. It's about their cognitive abilities. When I was watching penguins and other skuas, I felt that they sometimes recognize me individually, because I'm a invader and I'm an intruder to them. So when I went across the nest site, then I could see that they are a bit slightly moving their wings, and they are watching me a bit about 100 meters away from me, and then sometimes they try to escape from me. But when totally a new guy came there, they did not respond to the new guy, because they did not something bad to them. That means they can discriminate individual humans. Well, if you have a pet, then you can easily understand this kind of situation. But for wild animals, it's a totally different story, because for wild animals, they have no reason to distinguish individual humans. So there were almost no record about this kind of ability in the wild. So we did very simple test. Anyway, I got that kind of impression that they could recognize me, but I need to show this in a scientific way. So we made a pair of humans. One is neutral and one is the intruder. And then we separated into two directions. And then we recorded, we filmed the response of the skuas. And then I found that always the intruders were followed by the skuas. That means the flying birds were upset about the approaches of the nest intruder, and then followed them to make them out of the nest site. So I reported this case in the Animal Cognition Journal. I found this behavior in this very high cognitive species, brown skuas. And also I did very similar feeling from gentoo and chinstrap penguins, but I could not collect enough sample sizes. So I did not report this in the penguin. But recently I also reported similar behavior from Arctic birds. In the left, you can see that the previous studies were mostly in the domestic species, from dogs, sheep, cows, pigs, and they usually distinguish their feeders because the feeders are giving some good food to the animals. So that means the food is a positive stimuli. But in my case, it was a negative stimuli, and it was a negative stimulus, and so they try to escape me, or sometimes they are aggressive to the nest intruders. Yeah, so the third topic is the foraging behavior. So it's also another story. Sorry for confusing you, but my study is quite distributed to this kind of many topics. But actually, this is my main topic about the foraging behavior of penguins. So I'm very interested in their foraging site because the foraging behavior is highly related to the conservation. And Korea is the third country which are hunting the Antarctic krill. So many Korean commercial ships are visiting Antarctic oceans, and then they are catching a lot of krill, and then they are selling the krill to the people as a source of omega-3. So, and also instead of fish oil, krill oil became very popular in Korea. Of course, I don't eat the krill oil, but I'm very concerned about this kind of commercial fishing in Antarctica. So if we have this kind of data, then we can report our results to the governments and also to the international commissions. So based on my results, I found that penguins were amazingly foraging up to 70 kilometers away from their colony. 70 or 60 kilometers were really a long distance. To be honest, I was expecting that they could swim about 1 or 2 kilometers away from the colony, maybe, but they are very small, and they are about 50 centimeters, and they are weighing about 3 or 4 kilograms. But as a very small bird, they can swim about 60 kilometers for a day. That means they are really good swimmer, and they are foraging to the pelagic ocean. And also that means they have some ideas where are krill distributed. So I think they got some clues about the foraging spots. So if they have a knowledge about the hotspots of their foraging site, then they can easily go to the site, and then they can increase the foraging efficiency. And one of my recent publications is about their relationships between the species. From my title, it was indicated that I studied gentoo and chinstrap penguins at the same time. But it was because there were two penguins were breeding at the same colony. So if I go to the breeding colony, then there are two penguins in the same site. So they are totally different species, but they are quite similar to each other, and they don't care too much about each other. So they were not very aggressive, and somehow the breeding colonies were separated, but their breeding timings were quite similar, and their phenotypes are also similar. That means they will have very similar dietary habits. So you can expect that they will have a very high competition each other. But historically and evolutionarily, they would be somehow compromise. They had to compromise each other for this kind of competition. So it is called ecological niche. So people expected that chinstrap penguins and gentle penguins are similar to each other, but they had to find some way to reduce their competition. And then they find their own ecological niche each other. So I tracked chinstraps and gentle penguins at the same time. Then I found that their foraging areas were highly overlapped each other. But from the depth recorder, I found that they were a bit different vertically. So from the GPS locations, you can see that they were highly overlapped, chinstrap and gentle in Navarrete Point and Adelaide Island. In the two spots, the two penguins were overlapped, but vertically they were separated. That means their ecological niches were separated vertically, and they can focus on different foraging site. And this study was from the another station, Changbogo Station of Korean Research Station. It is on the opposite side of Antarctica. It is in the Ross Sea. In the Ross Sea area, about 5 years ago, something happened. It is called calving event. A large glacier was detached from the ice shelf. So from the satellite images, you can see that in 2014, there was some kind of crack. And then 2016, there was you can see that the crack is more clear. And then in the following years, it's totally gone. And the iceberg, and it has been a really big iceberg. And it floated to the north and north. And it was, so it was a big event. It was a big calving event. We have no idea why it happened. But probably it is related to the recent rapid changes in the Antarctica. I'm not a glaciologist, and I'm not an expert about climate changes, but this kind of rapid environmental changes are happening in the Antarctica quite recently. So one of this kind of big calving event could affect animals, I think. As a penguin biologist, I was interested in the result of this calving event. After this kind of big ice crack is created, then how the penguins respond to this kind of changes. Maybe you can expect that penguins will highly decreasing because of these changes, and then they can have less food. But on the opposite way, there would be more chances to go to the foraging site, and then this kind of calving event could expose more area, and it can provide more areas for fish and other benthic animals. So I deployed GPS and depth recorders to the Adelaide penguins in Inexpressible Island. Inexpressible Island is the closest colony from the Nansen ice shelf crack area. And then I found that among 27 individuals, 5 individuals were actively traveled to this area, to this newly exposed area, and then they did show some foraging behavior in this new side. So I think the second hypothesis is true. The second hypothesis was about that they could have more chances after this calving event. But as a short term, I don't know what it will eventually affect penguins in the future, but just for 1 or 2 years later, about after this calving event, penguins seem to be interested in this new area, and they seem to be foraging in this area. So I'm not sure about the effects of these calving events to the penguins, but anyway, I found that penguins were swimming in this area quite heavily, and still you can see that only 5 individuals went there among 27. That means it's a small proportion, but I want to know if this proportion is increasing in the future. So maybe 1 or 2 years later, more individuals can explore this area by the information changes. But at this moment, the 5 pioneers seem to be using this area. And as well as the GPS and depth recorder, I'm also using the video cameras to the penguins. On the left photograph, you can see that they were having something on their back. That's the video camera. And also on the head, I deployed accelerometers. Accelerometers are used to detect their head movement. So I was aiming to see that what kind of prey were attacked by the penguins, and also how they were interacting each other in the sea. Because for humans, we are very limited to watch this kind of diving birds, because humans are not able to dive deep like this. Penguins can dive up to 5-meter depths, 500-meter depths, but we humans can dive up to 5 or 6-meter depths only without any devices. So we are limited. So I deployed the video cameras and I filmed how they were doing in the sea, in the view of penguins. So based on the video cameras, I found that they were interacting each other very actively, and sometimes they made some communication call. So in the right picture, you can see that a neighboring gentle penguins is calling just in front of this deployed gentle penguin. So I found that when they were solitary foraging, they were more likely to make this communication call. Let me try to mimic the call, and then they make sound like a dog, like wah, wah, this kind of call on the sea surface. And then within 1 or 2 minutes, another call was heard, like wah, wah. Then they make wah, and then wah from the remote area. And then within 5 minutes, the neighboring penguins appeared in this recording. So I guess that they are communicating each other using this kind of vocal sound. And then if they are having some difficulties or they are having troubles to find food source, then they make sounds and they call other neighbors, and then they started to dive into certain directions, maybe for the food. Their main prey is Antarctic krill. So Antarctic krills were mostly moving together as a swarm, large swarm, like a fish. So for penguins, if they were moving in a group, then they were having more chances to find the food. So in a group, they need to divide the food each other, but it's a large krill swarm, so they will have only small. They have more advantages than their disadvantages to divide their food sources. So I think that that's why they are more likely to have to make this kind of group in the sea. And from the video recordings, I could clearly distinguish the species of krill. Sometimes they are Antarctic krill, and sometimes they are crystal krill, and also there are Antarctic cod, different fish species, and also there are squid in the Antarctica. So in the previous studies, people usually use the stomach flushing method. Stomach flushing means that after catching the penguins, you put the water into their stomach, and then you squeeze their stomach, and then when the penguins vomit, then you can collect the food from the liquid. Then you can collect it was krill, it was a fish. But using this method, it's highly biased because some of their prey are easily digested. For example, jellyfish is easily digested. So even if penguins were sometimes having jellyfish in the ocean, but it is not detected using this kind of stomach flushing. So I think video recordings is one of the direct method to distinguish their foraging diets. So currently I'm using many different kind of biologgers, GPS and depth recorders and video cameras, and also I'm using accelerometers to detect their head movements. You can even count the number of krill, because when they catch krill is about 4 or 5 centimeters long. It's like a shrimp size. So by one movement of head, they can catch only one krill. So if you count the number of head movements, that means that you can count the number of krill they ingested. So from this kind of combination of biologging techniques, I can very precisely calculate their energy requirement and their movement in the sea. So recently, I am very interested in developing a new method to sense their behavior. I am now trying to use another sensor, like using for example, like pressure sensors. So by deploying the pressure on the flippers of Antarctic seals, then maybe I can distinguish their behavior. I can categorize their behavior from their pressure signals. So when they are just resting on the ground, then there will be no signal, and then they were rolling, there will be a changes of pressure signals, and they are sliding or swimming, there will be a different signal. So using this kind of sensor, I am trying to figure out how they were moving in the sea. Also, sometimes it is very difficult to retrieve the signal, because biologgers are sometimes very difficult, because biologging is highly dependent on the retrieving. Retrieving means second catch after the deployment. But As I showed before, wild animals were very concerned about humans. So after the negative stimuli and negative experience, you got very sensitive to the same stimuli. So sometimes it is very, very difficult to catch the same animal for multiple times. So that means even if you deploy the logger to the animals, you may not be able to retrieve. You may not be collect the signal. You may just lose the devices. So in this case, I try to use the Bluetooth remote sensing method to collect the signals without retrieving them. So using Wi-Fi or Bluetooth, maybe you don't have to catch the same animal twice. You can just catch one time for deployment, and then you can select. You can collect the signal in a remote site in a remote way. Then you can sharply decrease the stress to the animals, and also you can increase the data efficiency. Okay, and the final topic is about the breeding ecology, and I'm very interested in the climate changes in the Antarctica, and in near the King Sejong Station, you can easily see that the climate change is ongoing. It's annually changing a lot, so you can visualize the changes. For example, in this map, you can see that the glacier boundaries are changing from 1989 to 2016. So I calculated the glacier retrieval rate, and the rate was about 10 meters a year. So since I first visited this area 2014, so it's been already 7 years. So for 7 years, about 70 meters of glacier already retrieved. So every year when I visit this area, then I can see that it's already gone. This glacier is already disappeared. So every year we have to map the coastline. So the map is changing annually. And also when you are working in the field, then you can hear the sound of this kind of glacier melting. Sometimes you can hear the sound of thunder, like quack, quack, like so. But there are no thunder in the Antarctica. It's always the daytime. But after the sound, when I spot this area, then there is a big glacier, and you can see the glacier melting. It's like a waterfall. So it's very sad to watch this kind of moment. But simultaneously, it became a new chance for some animals. Here, you can see the catgulls are breeding in this glacier retrieved area. So after glacier melting, a new ground is exposed. Then another animals can use this area for nesting. Catgulls are not they were not very common in Antarctica before, but quite recently they are expanding to the south. They were originally breeding near Chile and South Africa, but now they are easily found in King George Island, and there are new records of this kind of breeding expansion. So I think they got a new chance for a breeding site. But I don't believe that catgulls are increasing globally, but maybe I think their original breeding sites were affected by the warming. So I think that their distribution is changing to the south. And I just got the message from Dr. Chin that skewer are steady. And may I respond to the questions after my presentation? Maybe it'll be quickly checked.
Yes.
Yeah. Okay. So I have only a few slides. So I'm also using drones to watch the breeding colony. So in the Antarctica and Arctic, sometimes it's very hard to approach their breeding site because the breeding colonies are sometimes remote and sometimes blocked by iceberg and sometimes are very dangerous for human approaches. In that case, this kind of new method using an aerial devices can be very helpful. Also, in Antarctica, there are very cold background. So if you use thermal images, then it's more efficient to detect the warm objects, including humans. So sometimes they are very highly hidden and sometimes they are camouflaged. But using thermal drone images, you can easily distinguish their the number, and also sometimes you can easily distinguish their ages, like if they are adult or pups, or sometimes also for elephant seals, they are very highly skewed in terms of sexual dimorphism. So you can distinguish which one is female and which one is male from the drone images. And the final topic is about their gut microbiota. It's about their feces, like, because I didn't intend to study this area, but while I was staying there, I found that sometimes they were spontaneously fasting. They don't have anything for 2 or 3 weeks, and they are changing their feathers. For penguins, the feathers are very important for their survival. But while they were swimming, the feathers are sometimes the quality of feathers are decreasing. So every year they are changing their feathers. So they are changing their new clothing. So during that time, during this period, they are not able to go to the sea because it's too cold and they are not protected from the feathers. So they decided to spontaneously fast on the ground and they have nothing. So I could easily hypothesize that something's gonna change in their stomach and also in their intestine, because nothing is coming to the intestine. So in the view of bacteria, maybe they will have a very tough time without any nutrients. Well, for krill-eating species, maybe some bacteria is highly dependent on the krill nutrients. So I think that during the molting, there will be some kind of bacterial compositional changes. So I detected that in both species, in chinstrap and gentoo, their bacterial compositions changed. And also especially during the molting period, I found that some very interesting bacteria species. Some bacteria was producing some kind of sugar-like material. So I think it can somehow contribute to the host animals. It was previously reported in blue, little blue penguins and also king penguins, then that this kind of bacteria can contribute to their host survival, and during their fasting, they are totally have nothing. So some bacteria can affect the host survival, providing some sugar-like materials. So expanding my study about gut microbiota, I also compared the gut microbiome comparisons depending on the species. Some species are having grasses only. They are called herbivore. And some are insectivores. They are having insects only during the breeding season. So the three species were totally different, but two species are quite similar for the diet. So my hypothesis was that they are quite distant each other. The three species are distant each other, but if they are sharing their diet, then maybe they have more likely to share the bacterial compositions. So my result was similar with my previous expectation. So the snow bunting and sanderlings, the two insectivore species were sharing high bacteria, and pink-footed geese, which is a herbivore, have quite distant bacterial composition compared to other two herbivores. Also, I did a very similar study for the Antarctic seals. One is weather seals, and the another one is the elephant seals. So the two species were different each other, and I think this is basically based on their dietary composition. And for elephant seals are very deep diver. Elephant seals are sometimes going to 2,000 meter depths. So they are sometimes they are called the best swimmer. But compared to the elephant seals, weather seals are having about 500 or 600 meter depths, and their dietary compositions are a little bit different. So in this paper, I did not include their dietary composition, but probably this kind of dietary habits could be responsible for this kind of bacteria compositional differences between the two species. And recently, I'm doing another studies using biologgers. So as well as their behavior, I am also collecting the environmental signals. From seals, I could collect salinity or water temperature, and also from penguins, I am collecting their sleep signals from their head. And as a collaboration with French researcher about sleep behavior, we are preparing a study about reporting that they are even sleeping in the sea while they are on the surface. Sometimes they fell into sleep as a REM phase. REM phase means the rapid eye movement. That means penguins can also sleep with a dream. So and also I'm analyzing their fecal samples to detect their prey items. So using the feces, I can do many things, as well as the gut microbiota. So using many DNA techniques, I am now sequencing the prey prey species sequences and then matching these sequences to the library, and now I'm watching that which one was in the gut, which species was taken, and which one was contained in the feces. Yeah, so my talk is almost over now, and it's already 5 minutes ahead of 1 o'clock. So can I stop my presentation and respond to your questions? Yeah.
Yes, Dr. Wen.
Yeah, thank you for your attention. Thank you. So I think Dr. Chin previously questions about their cognition behavior. So yeah, I mentioned that there are two humans, and one is intruder and one is neutral. And okay, neutral, yeah, you're right. Neutral means that the human did nothing to the skewers, and sometimes they are new to the nest at least. So this neutral human could just pass the nest site, but at least this human did not survey their nest site, and they did not touch the nestlings. So that means this human is neutral. So that's why I named it neutral.
All right, thank you, Dr. Lee. Thank you.
Yeah, thank you. And the second question is about the collaboration of molecular study of penguins. Yeah, and yeah, sure. Our institute is quite open to the international collaboration, and we still have many international studies, and we are also very interested in collecting many samples globally. So yeah, our institute is having our own fundings for promoting this kind of international work. So Dr. Chin is accepted for this kind of international grant for exchanging researchers. So maybe we have many chances to do this.
Yeah, maybe I can add on this a little bit. This is called Corporate Asian Fellowship. I think it's open every year, and you can just log on to the website of Corporate, and sometimes the announcement is there, and you can apply. Yeah. Thank you.
Yeah, and also we have our Korean government is having some international funding programs. So maybe if you can find something from the Malaysia and Korea international program, then we can apply for the funding, maybe. But I don't know the details, but sometimes we are having some programs between European countries or also another East Asian countries. So probably we are having similar programs between the two countries.
Hi, doctor. Can I just ask a quick question?
Yeah, sure.
I wanted to know, like, because you study this behavioral ecology and all, so I wanted to know if is there any change of behavior, or like they tend to behave in a certain way because all this implementation of this stuff that you put on, like the GPS and the accelerometer and all this attachment. So do the penguins or like birds or anything else that you are researching on, do they behave in such a way that they feel like uncomfortable, and does that actually affect your results?
That's a very good question. So the early biologging researchers were very concerned about this matter because, yes, as you said, this device itself can affect the penguins. So I'm not saying that there is 0% of any effects of devices, but when we do some new devices, then we always have another control group. So we have one group is the deployed group, and the other one is not deployed and also using only dummy. We deploy the logger, and then we track their behavior, and also we track their breeding success. So we are very concerned about their foraging behavior, because it is our study purposes. But if it affects their foraging behavior, then we can fail our study purposes. So after comparisons, after multiple comparisons, we found that it is very little effects to the animals, but only biologging site researchers used very like large and very massive devices. So sometimes they used some kind of clothing to the penguins. And also sometimes they used some kind of belt on their belly. So I'm pretty sure that it could affect the penguins. But now we are using very slight and very small devices. It's really less than 1% of their mass. So I'm still trying to reduce the kind of possibilities of this device. Yeah.
Okay, thank you, Doctor.
Yeah, thank you.
Yes, myself. Hi, Doctor. So,
Hi.
may I know more on the foraging behavior of the animals in Antarctic regarding the food supply? Is it depending on the seasons? Or if yes, did you see more unfamiliar species foraging at your study site, which made the foraging activity increase?
Sorry, if I understand correctly, then are you your question is about their foraging items, or like about the seasonal changes?
Both. Is it influenced by the seasonal changes?
Yeah. Yes, there are seasonal changes, and for example, here I introduced two different periods. One is incubation and one is chick rearing. So before hatching, they are swimming more, and they can travel a longer distance, about up to 6 kilometers. So during this incubation period, they don't have to feed their chicks, because they can just incubate the eggs. That means they can just take nutrients for themselves only. So during this incubation period, it was reported that they were very, very less dependent on fish only. But after hatching, the prey items are dramatically changing, and then after the hatching, they are more dependent on the Antarctic krill. Because so we researchers are expecting that it is related to the feeding availability, because the Antarctic krill is more easier to feed the chicks, because chicks are only dependent on their parental feeding. So Antarctic krill is less digested in their stomach, and then they have more like lipid and also more like nutrients for the chick growth. So it was reported that that's why they are changing their prey items. But at the same time, I think it is also related to the environmental changes with the period, because incubation period is usually in October and November, and chick rearing period is in December and January. So with the seasonal changes, there would be some food availability will be changing for example, there could be some krill and fish are also moving in the ocean. So we at this moment, I have no evidence about that, but probably this kind of prey availability can be also affecting for this kind of seasonal changes.
which means like their incubation period and hatching period is fixed. Is it based on the seasonal changes?
You almost fixed, but still it's somehow changing annually, because in Antarctica as I said, environmental changes are quite rapidly happening now. So based on our long-term data, we are finding that it is more fluctuating. So I'm not saying that it is more like warming. It is more now it's becoming more unexpected. So with this kind of changes, I found that chinstrap penguins and giant penguins are somehow responding to these changes. So in some years they are hatching very early, but some years they are hatching late. But these changes are not very high so far. So yeah, somehow I can say it is fixed, because it is usually in December, but sometimes it is early December, but sometimes it is late December.
All right. Thank you, Doctor.
Yeah, thank you.
Dr. Lee, we have 2 more questions in the chat box. From Norasikin Hassan, have you done any study on birds other than penguins in Korea,
yeah.
maybe?
Yes. Yes, in Korea, I did some studies about black-billed magpies. Magpies are similar with crows. So magpies are very intelligent and like crows. So my PhD thesis was about this black-billed magpies, and it was about parental behavior. So at the time, I studied how the parents are allocating their resources to the chicks. It was about the sex ratio behavior. So my study was about how the parents are responding to the sex ratio of the chicks. So, for example, if there are many daughters and their only son, then the only son is favored by the parents, and in the opposite way, if there are many sons and there is only daughter, then the only daughter is favored by the parents. So it was a very basic question. It's happening in humans too. So I questioned about this to the magpies, and I found that yes, the rarer sex is favored by the parents. So as a result, in the natural variation, the sex ratio can be adjusted to the 1-to-1, 50-50. Yeah. So it was my PhD study. But now I became totally a new subject. Yeah. And I think, yeah. Sorry.
You have a second question from Dr. Hyo Chung-chi.
Yeah. So it's a very good question. I think the impacts are quite different to the species, penguin species. I mean that some Antarctic penguins are highly affected, and the population size is quite dramatically changing. So the most famous case is the emperor penguins. Emperor penguins have lost about 50% of their population size for the last 50 years. And the theoretical biologists are expecting that they can be extinct by 2100 if we do not reduce any greenhouse gases. And also, my study species, chinstrap penguins, is also decreasing in the Antarctic Peninsula, because chinstrap penguin is a krill. They are called krill specialists. So during the chick rearing period, they are very, very obsessed to the krill only. But gentoo penguin is different. They are called generalist. They can have krill, but sometimes they can have squid. And if there are no krill, then they can have fish. and squid and octopus sometimes. So now we are thinking that this kind of generalist strategy, penguins are expanding somehow compared to the other specialist penguins. So I am also expecting that there are more changes in the chinstrap and gentle penguin population sizes in the future. And, yeah.
I believe we have a question from Abdul Hasib. Do you want to ask a question?
Yeah, I want to ask a question from Doctor. Doctor, I'm from entomology, and I want to ask during your whole research career, are you find some insects like sucking lice, which feed on cow, goats, other animals in this region in Antarctica, or whole where present penguins? Are you find some insects on the body of penguins or not?
Yes. Yeah. It's a very good question, I think. Very, very occasionally, I found some kind of insects, like because I'm not have much knowledge about insects, but yeah, very sometimes I found one individual bugs from the penguin feathers. So probably it can have some feathers, but instead of animal bodies, I found more bugs under the rocks or near the penguin colony. From my presentation, you can see that on the left there are many like feces, and there are many rocks, and there are many red colored materials. They are the krill digested feces. So there are many like materials coming from the ocean delivered by the penguins. And also there are many carcasses of penguins. Annually, there are 5,000 breeding pairs are breeding, but many chicks are dying. But after 1 year, it's clean. So I expect that something is going there. Probably you guys are interested in this process. So insects are doing something. Also, I think microorganisms are doing something here for digesting the carcasses and for the nutritional cycling. But yeah, to be honest, I have not much knowledge about this. But still, I think that there would be a high potential for this study area, because previous Antarctic researchers were concerned about the large animals only. Otherwise, they were interested in the soil bacteria or something, not insects, because they believe that there are not many insect species in the Antarctica because it's too harsh for the insects in the winter season. But still, there are many insects, as Dr. Chin observed there. Maybe Dr. Chin can add some more description about this.
Yeah, maybe I can just add on a little bit about the insects that I found in King Sejong Station in Antarctica. So basically in the soil, as Dr. Lee mentioned, surrounding the nest, the feces, underneath the feces or the carcasses, you can see a lot of Collembola, the springtails. Those are the hexapods, the primitive insects. And we also found a lot of arthropods, such as the mites, the soil mites, the Oribarida, there's quite a number of them. But of course, in the soil, we will see other organisms as well, like the worms, the nematodes, the protozoa, the ciliates, and tardigrades, the water bears, and so on. Yeah, there are many living organisms underneath the soil. Yeah, and I believe what Dr. Lee mentioned, the bugs that on the body of the penguins, it could be parasitic arthropods like ticks. Yeah, ticks could be also found on penguins. All right, I think that's all my sharing. Thanks. Thank you.
Yes. Is there anyone else who would like to ask question?
No, thank you, Doctor. I'm very shocked to hear that there are a lot of species of insects present in that -degree temperature. Thank you, Doctor, both of you.
So if there's no more question, and can I ask one last question, Dr. Lee?
Yeah, sure.
I like the idea on the Bluetooth, but how far can you actually trace the animal back after the first deployment? And do the animals then not usually migrate way too far from one place to another?
You mean the migration behavior?
Yes.
Seasonally, yeah. Yeah, it's a very, interesting question, because I am also analyzing this data using another type of logger. It is called the geolocator. Geolocator is sensing the sunlight during the daytime and nighttime. So based on the length of this sunlight time, you can estimate the location as the latitude and longitude. But it's a but the accuracy is a bit high compared to the GPS location. But anyway, using this kind of geolocation signals, we are now analyzing their migration behavior, and we found that the migration distance was about 6,000 kilometers away from the colony. So it was amazingly quite distant from the colony, and sometimes they were separating into two directions. One is going to the Pacific Ocean and one is going to the Atlantic Ocean, and they came back at the same time, at the same period, to the colony back. And also, more interesting finding was that the pairs were divided into different directions. That means if the husband went to the west, then the wife went to the right. So east and west to the different direction, and then they came back and they meet each other next season. So I don't know why they were separating each other, but I think that the pair bonding is not quite strong during the winter season, and they were separating. And also, Pacific Ocean and Atlantic Ocean is providing very different environmental conditions, but still they are going to the different directions. And so I think we need to find out why they were separated. But yeah, they were very high migrants. So penguins should be called as a migratory birds. So in terms of this kind of migration behavior, but all the researchers did not have no idea about this. So this kind of geolocation techniques were applicable for about 10 years ago now. So now we are now approaching to their behavior more closely.
Thank you, Dr. Lee, for such a fascinating sharing.
Thank you.
If there are no more questions, I think that is all for today. Thank you, Dr. Lee. I hope that everyone enjoyed the talk and the discussion session, and don't forget to sign in the attendance form. Our next series will discuss on science policies with Dr. Megan Pimsley from National Institute of Justice, American Association for the Advancement of Science USA. It will be on 27 July 2021. So stay tuned with us for further notice. And on behalf of the organizer, we thank the presenters and thank you again. And remember to stay safe.